MHC-I molecule. So this is the part 1 of this particular video. In the second video we are going to discuss about the antigen processing and presentation by MHC class 2 molecules. So stay tuned and keep watching. So what are MHCs? MHCs we have already talked about that these are the major histocompatibility complex, right? That is the full form of MHC is, and these are basically the group of, there are certain group of genes which are present on the chromosome number 6, and these group of genes or the cluster of genes, they code for certain proteins which are found on the cell surface, and these are none other than the MHCs, right? So they help the immune system in recognizing the foreign substances, so whatever the antigens or the pathogens which are trying to invade inside our body, they help the immune system or our immune cells to recognize them, specifically the T cells. Okay, so they help the T cells in recognizing the different peptides of the different pathogens, right? Now there are two types of MHC molecule. We have discussed about that, the class 1 and class 2. Basically the cell surface molecules we are talking about. There is one more class is there, MHC class 3, but those are not cell surface molecules. They are mainly responsible for the cytokines and complement proteins production, right? So here we are not covering about them. We are talking about the MHC 1 and 2, which are mainly responsible for the antigen presentation and processing, right? So here in this particular video we are going to talk about the MHC class 1 presentation of the MHC-I expression take place in all the nucleated cells that we know about it, right? And they presents the peptides or the antigens to the T-cytotoxic cells, right? So this is very important that MHC-I molecules, those cells which are expressing the MHC-I molecules on their surface, they are presenting the peptides to whom? To the T-cytotoxic cells. So TC cells, the cytotoxic cells, they are going to recognize the peptides or the antigens when they are being presented by the MHC class 1. Okay, so mostly these peptides which are being presented by the class 1 molecules, they are endogenous or intracellular peptides, right? So intracellular pathogen, if some virus infected cell is there and that being processed by the cell and degraded into a small peptide, so those peptides will be presented by the MHC class 1 molecule. So mostly the endogenous peptides, although the exogenous is also being can also be presented by the class 1 during the cross reaction processes. Okay, so that I am not covering up here, right? Then MHC-I molecules are assembled in the ERs. Okay, so as we know that all the proteins mostly assembled in the endoplasmic reticulum, so MHC-I, since it is also a protein, so it is being assembled in the endoplasmic reticulum. Now we are going to cover up all these things in details with the help of a diagram, right? So now let's first talk about the antigen processing. Since we are discussing about the antigen processing and presentation, so there are two steps are taking place. One is the antigen processing, how the antigen is processed into a peptide, right? The processing means when a certain pathogen which is going to infect that is being degraded into a smaller peptides. So antigen processing we are going to cover, and next step we are going to talk about the antigen, that peptide presentation. Exactly, we can say peptide presentation instead of antigen presentation by MHC molecules, right? So antigen processing, talking about the antigen processing, if and this cell, let's suppose it is being infected by one endogenous pathogens, maybe some virus is getting infected the cell. So what will happen then? These endogenous antigen, let me zoom to you and explain to you. Let's zoom this cell and let's see if there is some virus just infect this cell, okay, and will release its component inside. So what will happen that this proteins which are released inside the cell, let's suppose if this is some immune cell or some healthy cell or any nucleated cell. So this is a nucleus, right? This is a nucleus. This is a endoplasmic reticulum, the rough endoplasmic reticulum. As you can see, ribosomes on its surface, right? So this is rough endoplasmic reticulum, and this is the Golgi apparatus, right? So this is the Golgi apparatus. So what will happen if this is virus infected? If this is a virus which has infected this cell, okay, and virus, we know that it always use the host cell machinery for application. So if this is the part of the virus which is being inserted inside, so what will happen? This part of the virus or the protein molecule, they are being degraded by means of a proteasome. Proteasome, you might be knowing that this is a barrel like structure which is actually a protein complex which degrade most of the proteins, some damaged proteins or the useless proteins which are not required by the cell. So the cell will consider this as useless because that is not the part of our body. So it will degrade, these proteasomes will degrade them into a smaller peptides, right? So it will degrade them into a smaller peptides, and these smaller peptides will then travel inside or translocates inside the rough endoplasmic reticulum by means of the certain TAP proteins. So TAP proteins are what? TAP is the transporter associated antigen presenting proteins, right? So they will help in translocation of these peptides inside the rough endoplasmic reticulum. And inside rough endoplasmic plasmic reticulum, I have also said that inside this there is the assembly of the class 1 MHC molecule will also take place, right? So let's see this. As you can see here, the assembly of MHC-1 has taken place, and when these peptides enter through the by means of the TAP protein inside the endoplasmic reticulum, they will go and bind with these MHC class 1 molecules on their peptide binding cleft, and then this MHC-1, which is coupled with the peptide molecule, or you can say the complex which is now formed between the MHC class 1 and the peptide, this will move out of the ER and will travel to the Golgi apparatus, right? And inside the Golgi apparatus, so let me show you here, right? So if this is the our MHC molecule from the Golgi apparatus, this will come out from as a form of secretory vesicles, right? So secretory vesicles will be formed, all right, and let's suppose this is a secretory vesicles. So here this is our class 1 with a peptide complex. This will move out on the surface of the cell and will express on the cell surface. So this is how the thing will take place. So I have told the presentation part also here only, okay? So this is the whole process is the antigen processing part, or the part of the viral proteins are being degraded by the proteasomes into a smaller peptides, and these peptides are then translocated into the rough endoplasmic reticulum by means of a TAP proteins, and then they couple it with the MHC-1 molecules, right? After these coupling, this will move to the Golgi apparatus, and from the Golgi apparatus this will then come out from this as a form of secretory vesicles, and these secretory vesicles then go out and release their inside content and express them on the surface. So this will then be expressed outside. So you can see here that you will find that like this kind of a structure. So this is a beta 2 microglobulin, right, and this is a alpha 1 2 3 domain, right, and this is the here the peptide is being presented here, right? So this is how the presentation of the peptide by the class 1 MHC molecule take place. Now this peptide is being then recognized by the T-cytotoxic T cells, which contain the T cell receptors on their surface, and these T cell receptors along with the CD8 co-receptor, they will recognize these MHC molecules in the peptides, right? So then they will generate immune response that this cell needs to be killed because it is a virus infected. This is how the interaction or the TC cells actually depend on the MHC-1 molecule presentation of the peptide. So this is how the antigen processing and presentation of the antigen or the peptides take place. So here if you look into the this diagram, this is also explained here. the same thing which I have already covered in the previous section. So this is a TC cell, or the CD8 positive T cells, and they generally have the GRAN enzymes. GRAN enzymes are basically the serine proteases which are present in the cytoplasmic granules, and they are being coupled with these perforins. Okay, right? So they are present in the cytoplasm of these TC cells and also the NK cells. So these natural killer cells and the TC cells are mainly responsible for recognizing all those peptides which are being presented by the MHC class 1 molecules, right? So if this is a virus infected cells, or a tumor, or a, you can say cells which is being tumor cell, or they are the target cell, you can say, right? So they are going to present a peptides with a coupled with the MHC class 1 molecules, right? And these peptides are then being recognized by the T cell receptors which are present on the CD8 cells or either on the NK cells, and they then along with the co-receptor CD8, they are going to activate these TC cells, and this activation of the TC cell then release all these cytoplasmic granules outside, and then these granules, cytoplasmic granules, since it contains the GRAN enzymes, which is a serine protease, which is an enzyme actually, this and the perforins, these, what it will do, perforins will create a pores in the membrane of the infected cell or the target cell, and due to the creation of these pores, these GRAN enzymes will enter inside this cell and will degrade the internal part of the cells, and there will be a lysis of this cell take place. So GRAN enzyme, basically what it is doing, it is inducing an apoptosis, right? Apoptosis, or the programmed cell death of this particular targeted cell. So that's how the whole process of targeting an infected cell in our body is being taken place by means of presentation of those peptides by the MHC1 to the either TC cells or to the natural killer cells. So this is all about the antigen presentation and processing. Okay, so thank you so much for watching. If you like my lesson, please do subscribe and share with all your friends, and let me know in the comment section down below if you have any queries related to this topic. See you in the next lesson. Bye for now.